In the Pacific, a drink made from a root could welcome a stranger, reconcile opponents, and affirm the authority of a chief. Centuries later, the same plant appeared in laboratories, capsules, and government warnings. How did a ceremonial substance become a test of modern herbal research, and can science judge Kava fairly without separating it from the people who preserved its meaning? The plant is Piper methysticum, a cultivated relative of black pepper whose name means intoxicating pepper. Its thick roots and underground stems are valued, while the leaves and other parts have not carried the same traditional role. Across island societies, knowledge of kava was not simply botanical information, it was a social inheritance, taught through practice, etiquette, and careful preparation. In Fiji, Yakona ceremonies have long accompanied hospitality, political meetings, and important communal occasions. In Tonga, kava has been associated with formal gatherings and rank. Samoa developed its own ceremonial customs, while Vanuatu contains diverse traditions in which kava drinking has carried social and ritual importance. These practices differ, but all warn against treating the Pacific as one uniform culture. The preparation was direct, yet highly meaningful. Kava roots were crushed, mixed with water, and strained or presented according to local custom. A decoction, meaning a preparation made by extracting plant material in water, sounds technical in a laboratory, but here the process also expressed trust, order, and belonging. The vessel, the sequence of serving, and the conduct of participants could matter as much as the root itself. European visitors recorded kava practices during the 1700s, carrying descriptions of Pacific ceremonies into expanding networks of navigation, trade, and natural history. Their accounts preserved valuable observations, but they also reflected foreign assumptions. A ceremony could be described as exotic while its political purpose went unrecognized. That tension would later reappear when researchers isolated chemistry from culture. Ethnobotany is the study of how people understand, use, classify, and value plants. Kava became an important subject within this field because its significance could not be reduced to a list of symptoms or ingredients. Ethnobotanical evidence asked a larger question. What happens when a plant's effects are inseparable from preparation, setting, relationship, and customary limits? The answer began to take chemical form in the 20th century. Researchers identified kavalactones, a family of phytochemicals found in kava. Phytochemicals are naturally occurring chemical substances produced by plants. These compounds became the main focus of investigations into kava's calming and muscle-relaxing effects, translating an ancestral remedy into the vocabulary of pharmacology. That translation was revealing, but incomplete. Different kava varieties contain different proportions of kavalactones. Growing conditions, plant parts, age, processing, and extraction methods can all alter the final preparation. A drink made from traditionally selected roots is not automatically equivalent to a concentrated commercial extract. The distinction would become crucial when kava travelled beyond the Pacific. In clinical research, kava was investigated chiefly for possible effects on anxiety and tension. Some studies suggested benefit, but the evidence varied in quality, preparation, and dosage. Such findings could justify further study, not sweeping promises. Traditional use may guide a research question. It does not by itself prove safety or effectiveness for every person or condition. The word adaptogen is sometimes applied to plants believed to help the body respond to stress. Yet it is a broad and debated term, not a guarantee of uniform action. Kava's best-known effects involve the nervous system, and its chemistry does not erase the need for careful clinical evaluation. The most responsible research keeps traditional knowledge and scientific uncertainty in view at the same time. Then the story took a darker turn. During the early 2000s, reports of liver injury associated with some Kava products led to warnings, restrictions, and bans in several countries. The reports demanded attention, especially because serious harm can be rare while still being medically important. But they also raised a difficult question. Were all Kava preparations being judged as though they were identical? Traditional Pacific beverages and modern extracts may differ in plant material, concentration, manufacturing, storage, and accompanying ingredients. Some products were assembled within unfamiliar regulatory systems, far from the cultural safeguards that surrounded customary use. This does not prove that traditional Kava is risk-free, nor does it prove that every commercial product was dangerous. It shows why categories matter. Scientists consequently examined possible mechanisms, including differences in extraction and the role of other compounds alongside kavalactones. Researchers also considered contamination, adulteration, individual susceptibility, interactions with medicines, and the condition of a person's liver. No single explanation resolves every report. Safety assessment is strongest when it admits complexity instead of choosing a convenient villain. For Pacific communities, however, the controversy was never merely chemical. Kava had become a symbol of cultural identity, hospitality, and continuity, while outside markets often treated it as an isolated active ingredient. A respectful look must therefore distinguish cultural practice from commercial marketing without romanticizing either one. Ceremony is not a clinical trial, but neither is a capsule the complete meaning of ceremony. This is where Kava shaped modern herbal research most profoundly. It demonstrated that a remedy arrives with a history of selection, preparation, language, and social regulation. Those details can influence exposure and behavior before a molecule reaches the bloodstream. In other words, the real object of study is not always the plant alone, but the relationship between plant, people, and practice. The lesson reaches beyond the Pacific. Willow bark helped inspire aspirin, and foxglove contributed to the history of cardiac medicines. Yet neither example permits traditional use to be copied without examination. Kava offers a different emphasis. The conditions surrounding a remedy can be part of its evidence. Ignoring those conditions may create a distorted experiment before analysis even begins. Modern researchers now seek more consistent descriptions of Kava varieties, plant parts, extraction methods, and adverse events. Such work cannot remove uncertainty, but it can make comparisons more honest. It can also encourage collaboration with Pacific scholars and communities whose knowledge should not be treated as a mine from which commercial science extracts value without recognition or return. The old ceremony thus survives in a new intellectual form. Its rhythm suggests that evidence is also a social act, requiring patience, boundaries, and respect. A hurried conclusion may isolate one compound while losing the larger pattern. A careful investigation asks who prepared the remedy, how it was made, who used it, and what the surrounding tradition understood about limits. Kava's journey from Pacific gathering places to modern laboratories is neither a simple triumph of ancient wisdom nor a warning against herbal medicine. It is a record of translation, misunderstanding, discovery, and correction. The kavalactones matter. So do the roots, the water, the varieties, the customs, and the communities that kept the knowledge alive. In the end, Kava asks modern medicine to become more precise, not less skeptical. It asks science to test traditional claims without stripping them of context, and it asks culture to be recognized as evidence about use rather than mistaken for proof of cure. Between ceremony and laboratory lies a responsibility shared by both. to pursue nature's possibilities without forgetting the human world that gives them meaning.